Okay, hello to all of you. Let's discuss chapter number 12. So we have some background about genetics. So we're going to continue with some more genetics. So let me go into slideshow mode. So basically in this chapter we're going to talk about sex-linked inheritance. So what is sex-linked inheritance? So in this, under the topic of sex-linked inheritance, we talk about the genes which are present on the sex chromosomes. So in the females, the sex chromosomes are 2X chromosomes. In the males, there are two chromosomes. One is X, another is Y. So in the females, there are 2X chromosomes, and in the males, 1X and 1Y. So basically, so in the females, because there are 2X chromosomes, and in male there is 1X and another Y, so the genes which are carried on the X chromosomes, they have two copies for the females, and for the males, because one chromosome is X, another is Y, so the gene which is present on the X chromosome, it does not have a copy on the other chromosome, because in the men there is only one and one Y. So if there is a mutation in the gene which is present on the X chromosome, in the men that disease is going to show up. Okay, suppose there is a mutation in a gene which is present on the X chromosome. In the men, the effect of that mutation will show up if there is a mutation. But in the case of females, if there is a mutation in the gene on the X chromosome, that the effect of that mutation will show up only if both the alleles on the two chromosomes, they are mutated. So what I mean to say is, in the females, because there are two copies of every gene, there are two alleles. So if one allele is mutated on one of the chromosome, it has a second copy. So in that case, the female, if there is one copy of the gene on one chromosome is mutated, that female is a carrier. Okay, so this person is a carrier of a mutant gene or mutant allele, but this female will be affected by some disease only if both the alleles are mutated. It will show up only if both the alleles are mutated. So in the case of man, because they have only one copy, if that allele gets mutated, the man will show up the disease. Okay, but in the case of female, because in the female there are two copies, if one of the alleles is mutated, that female is a carrier. She is not going to suffer from the disease, but she will suffer from the disease if both the copies get mutated. So here is one example of a disease which is because of a gene which is mutated on the X chromosome. So the disease is called Duchenne's muscular dystrophy. Is a X-linked disorder, because this disease is because of a mutation in a gene which is present on the X chromosome, and it mostly affects males. So Duchenne muscular dystrophy is an example of a X-linked disorder. This is X-linked trait. So recessive mutations of the dystrophin gene on the X chromosome cause the disease. Duchenne's muscular dystrophy primarily affects males because they inherit only one copy of the X chromosome from their mothers. Therefore, the single DMD allele they inherit determines their phenotype. Since females have two X chromosomes, they may carry the DMD allele but have a healthy phenotype. So here you can see there are two cases. So this is the father, okay, And we are going to do the Punnett scare the way we learned in the last chapter. This is the unaffected father, the normal father. These are the sex chromosomes X and Y. This is the mother, okay, and she is a carrier. So one allele on one chromosome is normal, another is mutated. So by meiosis, gametes will be formed. In the male, the gametes will be X and Y. In the female, one X. So this X chromosome has a normal allele of the dystrophin gene, and this X chromosome has a mutant allele for the dystrophin gene. And herely on this side, this is the unaffected mother and this is the affected father. So this is the unaffected mother. So both the chromosomes are normal, both the X chromosomes are normal, and this is the father, and this is the affected father. So that is why, which means the red means the allele is mutant here. So in the male, they have only one X chromosome, and if that if the allele on that X chromosomes get mutated, if that gets mutated, the disease is going to show up in the male. So these are the sperms formed by this man, and these are the eggs formed by the mother. So after fertilization, if this goes with this is a carrier daughter. If this goes with this, unaffected son. If this goes with this is carrier daughter, and if this goes with this is going to be a unaffected son. So on this slide is all the information I have already given, that females, they have two X chromosomes. So mother passes one of them, father passes his only X chromosome to his chromosome to the son. So that's all you need to know from sex-linked inheritance. The next kind of inheritance we're going to talk about is called incomplete dominance. So usually if you have studied Mendel's crosses in your previous classes, and I think I have also posted one video on Mendel's inheritance pattern. You go over it and see that. So according to Mendel, like if in a heterozygous situation, you know what heterozygous is, one allele is dominant, another is recessive, then the dominant allele is going to show up. Okay, so in the homozygous, no problem. If whatever the alleles are, they're going to show up. If both the alleles are dominant, the dominant character is going to show up. Both the alleles are recessive, recessive character is going to show up. But if one allele is dominant, another is recessive, so the phenotype will be of the dominant allele. So which means if one is dominant, another is recessive, one dominant, another recessive, then the dominant trait will show up, and recessive cannot express itself in the presence of the dominant allele. So what is, and on the basis of these Mendel did some experiments in his garden, and on the basis of that he formed a law of dominance, and on the basis of that he postulated that in the presence of the dominant alleles, recessive alleles do not show up. So this is a different kind of inheritance pattern. This is called non-Mendelian genetics. It's called incomplete dominance. So according to this inheritance pattern, so when the alleles, they are in the heterozygous situation, the character which is showed is not of the dominant trait, but it is something it is intermediate character or in between character. Like if you cross red flowers with white flowers, so when you cross red flowers with the white flowers, in the next generation you get the pink flowers. If this cross would have been following Mendel's inheritance pattern, then in the heterozygous situation, the color shown by the flower would have been red, because red is a dominant character. So similarly, this is showing. So if you take this heterozygous plant and make the gametes, and if you fertilize them, then you have here you have the red flowers, Then so this is homozygous, this is homozygous dominant, this is homozygous recessive, and this is heterozygous. So in the heterozygous situation, the color is pink. And here is the example of incomplete dominance um, of a disease. So in incomplete dominance, heterozygotes, as I said, display a phenotype intermediate between that of the either homozygous genotype. So we talk about this disease and we explain incomplete dominance in the disease called hypercholesteremia. So this is a disease in which people have a high cholesterol level in their blood. So the blood levels of the cholesterol are determined by the genotype of the LDL receptor gene. So this is LDL receptor gene. So this is a gene which codes for a receptor, and that receptor is called LDL receptor. The function of this receptor is the cholesterol from the blood actually binds on these receptors, and cholesterol is internalized and it goes into the cells. So LDL receptor normally helps clear LDL cholesterol from the blood. Okay. So people who are homozygous for the mutant allele have dangerously high levels of LDL cholesterol. So people who are homozygous for mutant allele, which means if this gene, this is a gene which codes for this receptor, if this gene which is coding for this receptor, and this receptor helps in, if this receptor helps in lowering the level of cholesterol in the blood. If this gene gets mutated, okay, what will happen then? Then this receptor is not going to be functional. If this receptor is not functional, people will have a lot of cholesterol in their blood. So people who are homozygous for the mutant allele, homozygous means people who have both the alleles which are mutated. People who are homozygous for the mutant allele have dangerously high levels of this cholesterol. Heterozygous have a moderately elevated levels of the cholesterol, and people who are homozygous for normal allele have normal cholesterol levels. So basically this is a gene which codes for this receptor, and this receptor helps in reducing the cholesterol level. So if both the alleles are normal, people have normal cholesterol level. If one of the allele is mutant, another is normal, one of the allele is mutant, another is normal, in that situation people have moderately elevated levels of cholesterol, because in this situation it shows some intermediate effect. And the people who have both alleles which are mutant, if the both alleles are mutant, then they will have dangerously high level of the cholesterol. So here is example of a case. This is the father with mild disease, which means one of the allele is mutant, another is normal. So here is again mother is also having the mild disease, which means one of the allele is normal, another is mutated. So here capital H is representing the mutant allele, and lowercase h is representing the normal allele. So you can make the gametes and you can fuse them. Okay, so you can see capital H goes with capital H. So it means that this child of these parents will have a severe disease. If this goes with this child will have mild disease. Again, this goes with this mild disease, and this will be normal. So this is basically showing what is incomplete dominance. Codominance. So codominance is a pattern of inheritance when the two alleles, when the two different alleles can both express themselves. So it is not that one of them is dominant, another is recessive. So if there is one allele is different, another allele is, if the two alleles are different, they both can express. That is called codominance. And the typical example of codominance is the blood type. So the people who have blood type A, so the two alleles of the two alleles for the type A, because this blood types, people like, you must be knowing about the blood types. People have blood type A, AB, B, or O. So it is all on the basis of the antigens which are expressed on the surface of red blood cells, and they are basically glycoproteins, and they're coded by the genes. So for the people who have blood type A, they have either both alleles A, or they have either one allele is A, another is O. And same thing for B. So they can be both alleles can be B, or it can be one is B, another is O. So but if the people who have blood type AB, they have two different alleles. Okay, so one allele is A, another is B, and in this case A and B both express themselves. So it's not that A is dominant or B is recessive. And this is called codominance. So a form of inheritance in which the effects of both are displayed in the phenotype of a heterozygote. So this basically shows that why it is so important to know the blood types. Okay, because when somebody needs the blood, when the blood transfusion has to take place, it's very important that the correct person gives the blood. Otherwise, if you get the blood from a wrong person, you can die. So during transfusion, it is very important to match who is giving you the blood, or who is the donor, who is the recipient. So basically, suppose this is a blood type. So the recipient is type A, Rh positive recipient. So Rh positive means there is one more antigen which is called Rh factor. So if Rh factor, so basically we have A, so we have blood type A, we have blood type B, we have blood type AB, we have blood type O. And then there is the presence of another antigen which is called Rh factor. If that is present, we call the blood type is A plus. And if this is present, then if somebody has a blood type B, we call the blood type is B plus. If this Rh factor, if Rh factor is not there, if it is missing, then we call it A negative or B negative. So it's very important to see who is the donor and who is the recipient. Suppose here is blood type, this is a recipient, type A, Rh positive. So the blood type A positive can get the blood from O negative, because O negative is a universal donor. Okay, and so blood type A can receive the blood from A can get from A positive, or they can also get from O negative. But if the person who has blood type A positive, and if this person receives the blood from, like, this is type B positive, this person can die. Okay, now how it is determined who can give blood to whom? So basically, the person who is the donor, it is important to see what kind of antigens their red blood cells have. And recipient, so for the recipient, it has to be seen what kind of antibodies they can make, because something is entering into the system. Okay, so when something foreign enters into the system, our body makes the antibodies against them. Suppose you are infected with some bugs, your body is going to make antibodies for that. So the donor's antigens and the recipient's antibodies are going to be checked. So if the donor is O negative is considered universal donor. So which means O negative means means they do not have any antigen on the surface, because blood type A means they have A antigens on them, B type means they have B antigens on them, AB means they have both A and B, and O means they do not have any antigens on their surface. Okay, so basically it is going to be checked on O negative. These people, they do not have any antigen on their red blood cells. So when they do not have any antigen on the blood cells, when that blood is going to enter into the body of the recipient, when that is going to enter into the body of the recipient, because there are no antigens on the surface of the red blood cells, their body is not going to make any antibodies. So that is why it is safe to give O negative. Now suppose the person who is A positive, suppose that person is getting the blood from B positive. So what's going to happen? Now B positive is a foreign to this person. So B positive means the blood has B antigen on the surface as well as Rh positive also. So when this blood is going to enter into the body of a person whose blood type is A positive, so B positive is a foreign antigen for this person. So the body of this person is going to make antibodies, and those antibodies are going to go and bind on the red blood cells of this donor, and these red blood cells are going to be clumped, or they're going to be aggregated, and because the red blood cells are getting clumped, the person can die. So it's very important that O negative is a universal donor and AB positive is a universal recipient. Polygenetic trait means there are some characters like human height or skin color. They are coded by multiple genes. Okay, so there is the involvement of multiple genes. A single trait determined by the interaction between alleles of more than one gene. So human height and skin color, both of them are the example of polygenetic inheritance, because height and skin are those characters which are coded by more than one gene. Multiple genes are involved. Like here for his example, for height, around 180 genes are involved. So here you can see this is showing the inheritance pattern of the skin color. You can see they're just showing here. I mean, for skin color, there is involvement of more than like again, it's just like more than around for skin also there are many genes, like around 150 or 160 genes are involved. And so here is just shown the three different genes which are involved, and you can see that these three genes are going to make different kinds of gametes, and these are the different shades of skin they are possible. The last one is multifactorial. So there are various characteristics which are not only because of the genes, but those characters are also determined by the environment. So there is an interplay of genes and the environment. So trait that depends on multiple genes combined with environmental influences are called multifactorial. So this is showing you an example of a flowers called hydrangea. So basically when they grow in basic soil, they show red color, and the same plant when they are grown in the acidic soil, so when these flowers, when they're grown in basic soil, which means the pH is alkaline, they show red color. The flowers show red color, and when they are grown in under acidic conditions, the color of the flower is blue. So this is called multifactorial. So here the plant is same, the flowers are same, the genes are same, but what is the difference? The difference is the environment. So this is a very typical example of multifactorial pattern of inheritance, and this tells us that it's not only the genes, it's the environment also which plays a very, very big role. So that's all for this chapter, and if you have any question, please let me know.